So thank you again. And we move to our next speaker, who is Joris Vermeesch, who is joining us from Leuven in Belgium, where he is professor of genetics. And he will be talking to us about mapping and sequencing the 22q11.2 low copy repeat rearrangements. Thanks on my behalf on the to the organizers of the 22q11 Utengsu Awards, because thanks to those, we started collaborating with Joris. We met at an earlier iteration of the symposium and then started collaborating. So, but not about me. Joris, please.
Okay, good evening or good morning, everyone. Thank you for the invitation to talk about our work here on mapping the low copy repeats. It's my pleasure to be here. And it has already been a pleasure listening to all of you and seeing some of you back, for which has been, unfortunately, been for too long that we didn't meet. Just as a reminder, and I think I don't need to tell you too much here, this is the structure of the 22q11 region. And what you see here, sorry, maybe there's a little bit too much information, but you see the blocks, you see A, B, C, D, E, F, G, H are the low copy repeats, which actually drive the rearrangements and which ultimately lead to deletion and duplication disorders in the 22q11 region. When we map this over different of our closest ancestors or closest relatives, actually, the pan, the gorilla, orangutan, and the macaque, you can see that actually these low copy repeats exist in those great apes as well, and not only the great apes and the macaque as well. So although the majority of the LCRs in the human genome we see a burst of the expansion of the low copy repeats during hominoid evolution, they are there already for a long time. And the overall structure of the genome has remained the same, in being that the order of the genes in the 22q11 region is the same in the pan, in the gorilla. There is quite a change in the orangutan. We see that there's a huge inversion, and within that inversion, there's a re-inversion of the C and the D block. So in the orangutan, we see quite substantial rearrangements, and in the macaque, we see more or less the structure as in human, except that there is an inversion between the C and the D blocks. So that's a little bit the overall broader overview of the 22q11 region. So overall, very conserved. The genes in the region are conserved, and even the organization of the low copy repeats. And it are these low copy repeats, of course, who drive the deletion and duplication. And just to remind you, how do they originate? Well, usually, we all have carried two alleles, two chromosomes, 22, and during meiosis, at least that's what we believe, is that during meiosis, occasionally, these low copy repeats can misalign across it, and then if a crossover occurs within this misaligned regions, then you generate a gamete with a deletion and another gamete with a duplication. And that's the main, let's say, idea that that's the way the genomic disorders arise, and also 22q deletion syndrome arises. What you also have to remember is that for every deletion carrier, there should also be a duplication carrier, because the gamete doesn't differentiate. That's not completely true. We see a little bit more deletions than we see more deletions than duplications if one starts screening neonates, and that's because we can also have an intrachromosomal or intrachromatid homologous recombination causing a deletion, and then you have some episome which get lost during further development, and therefore there's a little bit more patients with 22q11 deletion syndrome. Why don't we see more duplications? The phenotype is less penetrant and less prominent, and therefore duplication carriers or less often coming to clinic or genetic clinic. This was mentioned already and by Vermeesch. So if we zoom in, if we look at the 22q11 deletion syndrome, we only look at the rearrangements between these first four low copy repeats, low copy repeat A, B, C, and D. And if you look at and the reason we mostly look at those are because of the genes located in that. They're recognized either because some heart defect, and then the TBX1 or the crackle deletion causes is probably the major cause of these heart defects. It's not fully penetrant, but also some of the other phenotypes we know in 22q11 are driven by some of these genes. And so these are the deletions which we consider when we talking about the 22q11 deletion syndrome. The other rearrangements occur also, but we don't have proper frequency or in comparison. They are much less frequent as compared to the AD deletion. Okay. What is also important to know for all of you is that these low copy repeats are actually not just one block, one sequence which is repeated 8 times or present at these 4 blocks, but they are constituted of segments of 1,000 base pairs and more, which are present in different combinations, different orientation, and in the different blocks. And this is old work from the group of Vermeesch and also from Biennial and Tamim Sheikh, where they already in 2000 started to map or try to fiddle around with the organization of the low copy repeats and the segmental duplications and trying to understand how they were looking for. And the color codes show you which segments are coming back in different places. So the red block is present in A and B, and then you have the green block present in A, B, and D, and then you have blocks which are present in C and D, and so on. So, so they're patchworks of subunits which migrated over time, over evolutionary time. But if we look at the human reference sequence, there is still gaps in the human genome, and if you just look at LCR, at the LCRs A, B, C, and D, and look, we compared just the 2 latest reference genomes. The previous versions was 37 or HG19 and HG38, and there was some bigger major rearrangements here. And if you look at closer, just at LCR A, you see that there's 4 gaps. So a few years ago, we started wondering, shouldn't we look closer into those LCRs? We don't know what they mean. However, they seem to be somewhat conserved. And also we know very little about the genes in it. And this is a view of all the genes which are located here. And just by listening to Peter Campbell, I think it's interesting. This DGCR5 is right at the anchor point at the 5' end of the, or the telomeric end of the LCRA. Within this repetitive region, or a region which is at least repeated at some of the other LCRs as well. So I think it might be worthwhile to look closer into that indeed. So we set out to how is there a role for the LCRs in the phenotypic variability of the 22q11 deletion syndrome? We don't know, but if we would like to know, we need to understand the role of the LCRs. And therefore, we thought it's essential to close the gaps and sequence ultimately the LCRs. And of course, 20 years, this year, we're 20 years following the announcement that the human genome sequence was finished. Well, we know that that's was largely not true. I think now we're coming very close with the telomere to telomere consortium, filling the gaps, and also the Human Structural Variation Consortium, which is working on that. But short read sequencing has not allowed us to map anything of those variations. So we, now I'm going back a few years, let's say 5 years ago, we set out to close the gaps, and basically in the community we had two methodologies. We started from fiberfish, which is stretching DNA strands from individuals, and then we made probes, fish probes, for different subunits which were located in the LCR, so they were repeated, and you can see the repeats. But by having this color code, we know which fragments are repeated. And then by having long enough fibers and puzzling them together, we can basically reconstitute the whole region present on 22q11. In the meantime, last 5 years, there's another methodology been developed, bio-nano-optical mapping, which is also pulling fibers, and then with some restriction and modified restriction enzymes, putting fish probes on those DNA strands, and then based on the patterns of the long reads, of these long reads, you can reconstitute also where the fragments came from and reconstitute this region. Originally, this was the bio-nano-optical mapping very difficult because there was not a good reference, but if you have enough data and enough long reads, this becomes possible. And these two methods together have allowed us to really close in or zoom in on the region. And to our big surprise, maybe not big surprise, but to some surprise at least, we saw an incredible degree of variation going the region, and what you see here are in individual alleles. So a normal individual has 2 is diploid, but this is the sum. We mapped 44 of those alleles, and you see that the region is extremely variable. We didn't see any individual who was homozygous for the LCR22A. And moreover, what you should appreciate is that the minimal size here was about 300 kb, and then the largest is 2 megabases in size. So this is really a huge block in that region. And LCRD, 22D, you see a little bit of variation, but a little bit less. We have an inversion, which is quite frequent in the population of 64 kb here, and then some more rearrangements here. And we, and also the team of Bev Emmanuel, has mainly Bev, has been looking also at the variation in the population, and has shown that that some of those variants which we see in the LCR 22A are more frequent in certain populations as compared to other populations. Okay, that's just the observation. So another question we were asking was, is this 22q11 variability? Well, following this observation of this variability and also this population variation, or this variation within the human population, we wondered, is this variability something which is also present in the great apes, or what? How do these LCRs look like in the great apes? I showed earlier that they were there, that there is some type of LCR, but the shape of these LCRs has never been mapped. So we set out to map this variability in PAN. So we looked at the bonobo and the chimp, then the gorilla, orangutan, and then macaque. And basically what you can see is that in PAN, in chimp, you have a very short unit which only exists out of this first block, in which prod H and then the DGCR5 is located, and then some region which is recurring, and then the anchor, the other anchor. So basically the 2 anchors are maintained. This allele, we've seen it in human, but only I think 1 or twice in the paper of Bev, and definitely not in homozygous form. So if you look at the gorilla, we have some other units which are somewhat rearranged and not really the same sequence anymore. And then in the orangutan, there is some variability, but the variability is different as compared to the human. And macaque more or less is this shortest unit, which we see also in chimp. So here we see that the expansion, or at least this extreme variability, is human specific. So then that raises the question, is there a meaning? Does it have a role? Has it phenotypic effect? And or the other hypothesis is, of course, it's just genetic drift, because once it starts to become longer, it starts to affect the expression in the region, and maybe it doesn't mean anything. But at the same time, the other question unanswered, which remains unanswered, is why is 22q11.2 deletion syndrome the most frequent genomic disorder? If you remember, the AD deletion is present in 90% of the cases, but the 22q11 disorder is about 10 times more frequent as the second most frequent genomic disorder. And therefore, which is probably an F1 deletion. And so what is driving this? And maybe it's the size, maybe it's something else. So to address this question, we really thought that mapping and sequencing 22q11 Lcrs and the rearrangements would provide the answers. And now I think there's two technologies with long-read sequencing, which actually opened this door. And just, I'm not going to go into that, but basically we have the SeqPac BioSequel, and maybe even more exciting is the Oxford Nanopore PromethION. And the reason I say it's more exciting is because the reads we get are maybe not perfect in sequence, but they're longer. And the fact that it becomes feasible, I referred already to the structural variation consortium and the telomere to telomere consortia, who now have a number of papers out. The first full-length human X chromosome was sequenced by the consortium, and then there was earlier this year the first full-length chromosome 8 telomere to telomere sequenced. Note that is just a single chromosome in a hydatidform mole, which is haploid. So this is not a diploid individual. So, but nevertheless, it allowed for the first time to go from telomere to telomere. And I zoom a little bit in on this chromosome 8. Why I got excited about that is that in chromosome 8, in the human reference genome, there were still 5 gaps. And basically, by having ultra-long read ONT sequencing and maybe a little bit shorter reads, PacBio HiFi and ONTs, and then integrate that together, they could basically close all the gaps. And in addition, well, what did they discover? They found new genes. They found new epigenetic patterns. I should mention that with ONT, we can also map the epigenetic patterns. And they found some new features in the evolution. And if we zoom in more, on chromosome 8, we have the...
Joris, about 2 minutes warning, please. 2 to 3 minutes. 2 to 3 minutes.
Okay. Thanks for mentioning this, because I'm really spending too much time.
I love seeing it. I hate to jump in. Sorry.
Okay. No, thanks for saying that. Okay, so I'll just move on what we have been doing. First, we scaffold with this fiberfish. Doing so, we can map breakpoints up to 20 kb regions. But then we move on. And from here, either the fiberfish or the ONT shows that there's some rearrangement for which we can generate a long PCR, 10 kb fragment. We sequence that fragment using long-read sequencing, and we can map the location of the breakpoint. And in this particular family, we identified that the breakpoint should be around these red blocks or PATR sequences. Here we didn't have the parents because it was familial, so the grandparents died already. So we're not completely sure exactly where the breakpoint is. But then we started doing long-read sequencing, ultra long-read sequencing, where we have N50 of 75 kb. reads up to a few megabases. This one was 1 megabase, but now we have even longer. And then we can actually get to the breakpoints. And I just bring you this particular family. It's an individual who came in for non-invasive prenatal testing. And actually, we discovered that she was carrying a 22q11 deletion. The fetus was normal, but she was a mosaic actually for 22q11. We mapped both alleles. It was 50-50 the deletion, and in 50% of the cells, she carried an inversion. So this is allele 1, allele 2, allele 3 is the inversion, and then we had also a deletion. So we did this ultra long read sequencing, and so we can now map actually where the LCR A is. And what you're seeing here is the locus in red are the AT-rich regions, which actually contain the palindromic regions. So the AT-rich regions are palindromic. And these palindromes are likely, are known to be subjectives to cutting. So it's likely that they're cut, and then they can drive rearrangements. And that's what's shown for the 22q11, the translocation 1122. So in this family, we also got the mother of this future mother. And basically, I'm going to go really fast just because I realized I was spending too much time in the intro. What we found was that there's actually first an inversion has happened probably during meiosis. We can call it meiotic recombination or meiotic break in a PATRR. We can really zoom into that. And then we generate a mitotic recombination in this particular case. I think mitotic recombinations are very rare in 22q11. But it's still challenging or impossible with ultra-long reads. You have to be a little bit lucky to really cover the whole region. So we focused on Cas9 mediated cutting and targeted sequencing. What we basically do is generate Cas9 probes, probes cutting within the LCRs and flanking the LCRs, cutting those out with those enzymes, and then you can actually targeted resequence those regions. And this is an example where we enriched the locus for about 60-70 times and had reads going over the breakpoint. In this particular case, the rearrangement occurred not in the PATTR, but in some other repeat region. And then finally, we collaborate with a group of Stanford, Alex, or actually this is a technology developed by Alex and his team in Bozu, a CRISPR-targeted long-read sequencing, which is similar to what I described, except that for the longer fragments, they're first separated on gel electrophoresis, pulse gel electrophoresis, cut out and then resequenced, which allows us to really capture all possible regions. And I hope Leuven is going to join the team there to finish that. And so that's where we are at the moment. So I think what it looks like is that in addition to the traditional non-allelic homologous recombination, we have some PATRR mediated mechanism which drives these rearrangements. I think we need to confirm, and we're now really upscaling this approach to map a large number of samples. We're improving the software to allow diploid haplotype resolved mapping. And then we want to zoom in just the LCRs, not only of the patients, but also the population. And this is done by Nicolas, postdoc in the lab, who's actually developing the software to do that. And I'm not going to zoom into that, but compared to the current best method, Shasta, is moving ahead fast. And I think we can now read through these regions. And then I think the next question we can then start addressing is, can this LCR variability explain some of the phenotypic variability? And this will come from large numbers of patients where we can do long read sequencing. And we also require those haplotype resolved assemblers. And then last but not least, I think we need the sequences in order to understand the sequence expression of genes, the genes located in those regions, and we can use the variation present in these LCRs A, B, C, D to allocate which paralogs are expressed and which ones may make the difference there.
Joris, we have to wrap up, I'm afraid. Sorry,
No,
I could spend hours on this, but sorry.
It's fine. I apologize if I went too long. I just would like to thank mainly Lisanne, who's really the PhD work I was presenting. Ruben is a new student in the lab. And of course, I need to thank also Bev and Donna and Ambassador for providing me and us families to work on this and map all those regions. Thank you.
Fantastic. Thank you very much. Very exciting. And we already have lots of questions. We're holding them.